Welcome to Canine Cancer Alliance webinar. So my name is Mary Maeda. I'm the founder of Canine Cancer Alliance. Canine Cancer Alliance's mission is to support research, fund research, so that we have much better, safer, effective, and affordable cancer treatments for dogs. And I know it is possible to do that. We're also interested in prevention and strategies to reduce risks of cancer as well. Today, we have as our guest Dr. Catherine Wise from Duke University. And Dr. Wise is an expert in environmental chemical exposure for animals and humans. And we first found out about her work earlier this year. Actually, we should have known about your work earlier, but we came across your paper, a really exciting publication about a study they had done with cancer, with bladder cancer in dogs, and linking it with various environmental exposure. And we started having discussions about possibly applying some of the things, approaches they're using to understand why there are so many dogs getting hemangiosarcoma. And I probably don't have to explain to this crowd why hemangiosarcoma is so important, but hemangiosarcoma is one of the most common in dogs. There is an equivalent cancer in people called angiosarcoma, and there has actually been incidents of environmental chemical exposure, workplace chemical exposure in a factory for people where this very, very rare cancer ended up occurring in several of these workers at the workplace. So we know that there might be some chemical exposure linked to elevating the risk of getting this type of cancer. But I'll let Catherine describe what she's going to be doing in this new Canine Cancer Alliance sponsored study, but also describe some of the other exciting work that she has done with other types of cancer using the techniques that she has. So let Catherine take over. And so Catherine Wise from Duke University. Thank you.
So thank you for the introduction, Mari. So today I'm going to talk about dogs as sentinels of exposure and health effects. And so just a brief overview of what I'm going to talk about today is this concept of dogs as a sentinel for human health, a little bit of just what environmental exposures are, how exposures impact dogs' health, and I'm going to tell you about the bladder cancer study that we did, and then talk to you a little bit about our current research, which is focused on hemangiosarcoma with the Golden Retriever lifetime study in the Canine Cancer Alliance group and the Dog Aging Project. And then just a little bit about what you can do as dog owners. So sentinels. The word sentinel has been referred to as somebody who keeps watch. And as our colleague, Dr. Audrey Ruppel said, that this is what dogs have always done from the beginning of time. Guardians of our livestock, of our families, and of our homes. So it's this concept of, like the canary in the coal mine, where canaries were brought into the coal to warn miners of toxic exposure. We have the canine on the couch. So can dogs inform us about health hazards in our own everyday environment? Now, there are hundreds of shared diseases and chronic health conditions between dogs and people. And this is just an example of a few of them. And our research is mostly focused on cancer. And there are a lot of cancers that are shared between dogs and people. And by shared, I mean they have the same clinical presentations and clinical management. A lot of the tumors look both large, on a macroscopic and a microscopic level, they look very similar. And there's a lot of genomic and chromosomal and molecular alterations that are very similar or the same in both species. The cancers often have the same, will have the same origin and have the same sites of metastasis. And we use the same types of drugs to treat them. But what else we have in common is this shared environment. So we have this shared disease, and what we want to know is this shared environment, are the factors in our environment contributing to these shared cancers? And why dogs? Well, first of all, a lot of us here today because we care deeply about our dogs, and we want to know better ways to help them and keep them healthy and protect them. But dogs can also be a good model for humans. And why is this? Well, dogs don't smoke and they don't have these occupational exposures, which are some of the biggest confounding factors. So the things that make it difficult in human studies to understand relationships between exposures and health outcomes. Dogs generally have a more consistent diet, so there's less variable there. They also have a robust medical history that is recorded, and we're able to look over their medical history over their lifespan. Time is the other critical thing. For a lot of chronic conditions like cancer, it takes decades for a cancer to develop in people, whereas in dogs, this is on a scale of 1 to 2 years. Now, a group actually did this study where they showed that there's this conserved aging clock in dogs and people. So what we mean by that is that there's this relationship between when dogs reach major physiological milestones, and showing that there's this nonlinear relationship between these milestones and aging in dogs and people. And when we talk about cancer, when you account for that that relationship, you can look at the time period and the life stage in which most cancer deaths occur. And this is occurring in dogs and people about the same life stage. An important thing to consider with cancers or other chronic diseases is that genetics are only a small part of the picture. A lot of the factors that are associated with chronic diseases are actually environmental factors. And what we mean by environmental factors include things such as nutrition, which can be vitamin deficiencies, high-fat diet, lifestyle factors like lack of exercise and obesity, and inadequate sleep, and tobacco use. And then there's environmental chemical exposures. And this is the area where most of my research focuses. So what are environmental exposures? Well, you may recognize some of these older headlines talking about lead in gasoline or paint, asbestos, DDT, industrial pollutions. And a lot of these are symptoms or are things that we thought were safe, and we just didn't know much about them, or we didn't think much about them. But we learned that they have lasting effects on human, wildlife, and now pet health. And a lot of these things have shaped the foundation for what environmental safety and public health regulations are today. But fast forward to today, in an era of instant communication and global data sharing, more advances in science and technology, we can now detect chemicals like PFAS and microplastics sooner. And we can identify these emerging issues faster and hopefully act sooner. So a lot of these exposures aren't just history. A lot of the chemicals of the past are still persistent in our environment. And in reality, we are exposed to thousands of chemicals each day. But as we look at these headlines and we think how many things are happening, how many chemicals there are today, we're not focused on, the fear of it, or we're focused on this story of progress and awareness and accountability, and how much we have learned and how much we can do now with this massive communication and that we have globally to protect both people and our pets from the emerging contaminants in the environment. And some of the things that you may have heard about are heavy metals such as lead, like in Flint, Michigan, or these forever chemicals, the poly and perfluoroalkyl substances, or PFAS, which are found in a lot of nonstick cookware and used in a lot of things that have, water or stain repellency. There are pesticides. The big one today seems to be glyphosate, whereas in the past it was DDT. Plastics. This is both microplastics, so these tiny particles of plastics that have been detected, in our bodies and, in our ecosystems, as well as the chemical plastic additives that get added to plastic, some of which you may recognize as like BPA, which a lot of things are now advertised as being BPA free. There's persistent organic pollutants. These are some of those chemicals that used to be used, and now we've learned that they're not the greatest for health, so they've been banned in their use, but they're still persistent, and we can still detect them in the environment and in biological tissues. Then there are volatile organic chemicals. You may have heard this from that train derailment in Ohio and the concerns about the chemical exposures from that. And then there's just the big question of air pollution, which today you hear a lot about, both from the industrial sources, but also from wildfire smoke, which is very prominent in our world today. But a lot of these chemicals are, in our everyday environment. We're concerned about a lot of indoor air. So a lot of these chemicals are in building materials and common household items, and that's how a lot of us are exposed because we spend so much time indoors. So these can be things like pesticides, which we apply to our lawns, but also directly onto our pets, in building and insulation materials, in our personal care products, in things like flame retardants or PFAS chemicals that are used in stain repellency in furniture, as well as things that are used in electronics, and also things like these plastic chemicals and adhesives that can be in different types of flooring. But how do we test for these exposures? How do we test our pets for these exposures? Well, there's two major ways that you can do it. First of all, you're looking at the external. So what is in the environment? And this can include things like what's in the drinking water or the food, but also what's in the dust or the air. And then I'll talk about this a little bit more later, but we are using this silicone personal monitoring device to be able to determine what type of chemicals are in the ambient environment. The alternative is to look at internal exposure. So this is actually measuring what's in the body. So we can look at blood, nail, hairs, urine, and feces for this. For exposure monitoring in dogs and in people, what's typically the gold standard is looking at blood and urine. These are readily available biospecimens that people use to measure exposures. But with our dogs, this can be challenging because it often requires a trip to the vet, which can be stressful for our dogs, and it can be expensive and time consuming. And also, it can be difficult to chase your dog around the yard trying to collect a urine sample. So we use this tool called silicone samplers, and these were originally used and introduced in 2014 as silicone wristbands to measure exposures in people. But they've also been used in cats and dogs, and they capture these semi-volatile chemicals from the ambient environment. So we're able to just attach these to a dog's collar or have people wear them on their wristbands, and you wear them for a set amount of time, and then they can just be shipped back to the laboratory where we process them and measure the chemicals that are detected in them. And these are really good for capturing the ambient environment. So that's things that are in the particles, so the things that are in the dust, as well as inhalation. But there's potential for some things that are either on the skin, such as lotions, or on the fur, such as those flea and tick preventatives that we put that could also get onto the tag, and we can measure those exposures. And importantly, these chemicals, what we're measuring on them, first of all, we showed that people and dogs living in the same house, this first graph on the upper portion is just showing that this chemical permethrin, which is a common pesticide, is significantly correlated between people and dogs. So that means dogs and people have very similar exposures to this chemical. And then if you look on the bottom, these two are showing the urinary biomarker of this chemical compared to the chemical on the wristband. And what this data is showing you is that what we're measuring on the wristband is correlating to what is detected in the urine, which means that what we're capturing is very relevant to what's actually getting in the body. Now, how do environmental chemicals impact dogs' health? There's a whole ton of evidence that has been studied so far. And just to name a few, cancers, there's a lot of different cancers, but the three that have been the biggest focus and that there's the most data about currently in terms of environmental exposures is bladder cancer, lymphoma, and mammary cancer. And the other big ones are kidney disease and endocrine disorders such as hypothyroidism and reproductive issues and fertility, aging, and then there's others, but much fewer studies have been done. And this QR code is just a link to an article that we published that just has the full summary and details for all of the exposures that have been identified in association with these diseases. So the big study that we did was bladder cancer. So bladder cancer, 90% of bladder cancer in dogs is urothelial carcinoma, which has previously been referred to as transitional carcinoma, and you may recognize as the term TCC. It is a malignant cancer of cells that are lining the bladder. These are the urothelial cells, and 85% of dogs that have urothelial carcinoma have a single mutation in a single gene. Bladder cancer is found in the urinary tract, so it's mostly in the bladder, but it can also be in the urethra, in the prostate, and in the up into some of the parts of the kidneys. And often at times when the dogs are diagnosed, it is already an advanced stage disease, and they already have tumors that are invading into the muscle tissue and have metastasized into local lymph nodes and sometimes even further into the lungs or the liver. There's about 60,000 new cases of dogs in the US that develop bladder cancer. And the most common breeds that are affected are West Highland White Terriers, Scottish Terriers, Beagles, Shetland Sheepdogs, and it is more common in female dogs. Now, it's primarily diagnosed later in life, in dogs that are middle-aged or older, and 95% of them are over the age of 6. But the big question that we're interested in is why? Well, there's no single cause. Genetics and the environment are likely working together to increase the risk. And what a lot of people have heard over the past decades is this has become this dogma in veterinary medicine that lawn herbicides and pesticides increase risk. And this has all been based on questionnaire-based data that dates back to the 1980s. But other studies that have linked bladder cancer to specific pesticides and herbicides, just general air pollution and industrial activity, secondhand cigarette smoke, and disinfection byproducts in drinking water. So those are when chlorine is used to disinfect water, it can react with the organic material and create these disinfection byproducts such as trihalomethanes. And I'm just going to highlight just 2 of the most recent studies. So there's a group, Knapp et al., that published a study in 2004 where they showed specifically Scottish Terriers that come from households that smoked were 6 times more likely to develop bladder cancer compared to dogs that don't live in smoking households. And then this figure that we're showing here is just showing that although they didn't have this statistical significance, there was a 10 median pack years of exposure in dogs from smoking households compared to people that weren't from non-smoking households, only had 1.5 pack years. And some of the confounding in this that makes it not quite statistically significant is the complication of people that previously smoked, but they stopped during the duration of their dog's lifetime. And then another study, which has been done once before and didn't quite have statistical significance in their findings, but this most recent study found that those disinfection byproducts, the trihalomethanes, dogs with bladder cancer had 4 times higher levels of these disinfection byproducts in the municipal drinking water compared to healthy dogs. So our study, what we did, we partnered with NC State Veterinary Medicine and Dr. Matthew Breen, and we did this study funded by the V Foundation, looking at high-risk non-muscle invasive bladder cancer and trying to look at dogs that are at risk for bladder cancer before it's clinically manifested. And we looked at both these molecular and environmental considerations that impact risk and development of bladder cancer. So we had 101 dogs from across the United States that were participated between 2018 and 2022. And we had the participants have their dog wear these silicone tags on their collar for 5 days. And then we tested these tags for 115 different chemicals. And then we compared dogs that have this specific BRAF mutation that I mentioned to dogs that are age, breed, and sex matched without the mutation, with the goal of trying to determine whether any chemical exposures were found more often in dogs that are at risk of developing bladder cancer. So as I mentioned, we looked specifically at 115 chemicals, of which 39 chemicals were detected in greater than 50% of our population. And this is just a breakdown of the different number of chemicals from each chemical class, which are organic phosphate esters, polycyclic aromatic hydrocarbons or PAHs, PCBs, pesticides, phthalates, and brominated flame retardants. So the goal of this is to say, is the chemical present in our samples? If so, how much? And is there more of it in one group versus the other? So our cases versus controls. And we found that there were 4 chemicals that were statistically significantly higher in cases. So the first one on the upper left is this combustion byproduct anthracene, which is a PAH. A plasticizer, benzyl butyl phthalate, which is BBP, which can be found in vinyl flooring, and then 2 flame retardants on the bottom, which are both PBDEs, which were used as flame retardants historically, but have been banned for use, but still persist in some of the products that we have in our homes. So the next thing that we asked was, well, what other chemicals are present in our samples? So I'm just going to introduce you to the plot that I'm going to show. So this is a volcano plot. And on this graph, each dot represents a single chemical feature. So think of each dot as one chemical. And if you look on the vertical or y-axis, we have the level of significance. So anything that is above those two, pink or red lines is significantly higher in group 1 or 2. And so anything to the right would be higher in dogs on that side, and anything to the left would be higher in the dogs that are on condition on the left side. And then across the x-axis, so horizontal, the magnitude of change between the two conditions. So if it's farther on the right and higher up on the right, there is more of it, and it's more significantly different than what's the condition on the left. So here's our data. Now, here's that midline that we're talking about. So that would be any points that fall there would be equal between the controls and the cases. Our cases are on the right and the controls are on the left. And what I hope you can appreciate from this is simply that there are a lot more chemicals on the right side of the graph. So the cases seem to be exposed to a higher number and greater amounts of those chemicals. So they have a higher exposure burden compared to the controls. So our 2 big takeaways were that dogs with the BRAF mutation are exposed to more chemicals and higher levels than dogs without the mutation. And the specific chemicals, including 2 flame retardants, benzyl butyl phthalate and anthracene, were significantly higher in dogs. And then here's just the QR code that's just showing you the full article that we published for our findings. So what are we currently working on? Well, big project that I think a lot of you are here to hear about is the work we're doing with the Canine Cancer Alliance and the Golden Retriever Lifetime Study. So the Golden Retriever Lifetime Study is a nationwide study that started enrolling dogs in 2012, and they have just over 3,000 Golden Retrievers from all these different areas of the US that enrolled when their dogs were mostly puppies, and they observed these dogs over their lifetime. So the goal was they wanted to identify the nutritional, environmental, and lifestyle and genetic risk factors that contribute to cancer and other diseases in dogs. And by doing this, they've established a large database which has annual surveys that are conducted by the owners, annual veterinary exam records and surveys. They have a biobank which has annual collections of a variety of biological samples from these dogs each year of their life. And then every year they open up these applications for researchers such as us at Duke to propose studies to explore critical questions related to the diseases that have developed in these dogs. So we're doing a project with this for hemangiosarcoma, which is the most common cancer diagnosis that these golden retrievers have. Hemangiosarcoma is a common and aggressive cancer of the blood cells or the endothelial cells, and it can occur in two places. One is the cutaneous, so in skin, or visceral. And the visceral is most often in the spleen, heart, or liver. Golden retrievers, German shepherds, Labrador retrievers are some of the most at-risk breeds. And when it hits, it's usually a pretty devastating disease because there's often no warning signs, and it's often diagnosed really late. And the prognosis and survival time is not great. But why does this happen? That is the big question that we're all wondering. And the answer is it's not really understood. There's still a lot of work that's going into understanding why these dogs develop hemangiosarcoma and why it's diagnosed so late in their life cycle and in their disease progress. And in the golden retriever study, there is just showing you how devastating this visceral hemangiosarcoma is. This figure is showing that just the poor survivability after dogs are diagnosed. And from this, the types of visceral hemangiosarcoma are predominated by the splenic hemangiosarcoma. So that's what we're focusing our efforts on, is looking at the dogs specifically that have developed hemangiosarcoma in the spleen. So our current study, we have 300 dogs that we're enrolling in this study, and these are dogs from the Golden Retriever Lifetime Study. So we are getting 100 dogs that have been diagnosed with splenic hemangiosarcoma, and we're comparing them to 200 healthy sex and age-matched controls. And importantly, we're getting blood serum from these dogs to measure chemicals in prior to their diagnosis. And we're looking at some of these persistent organic pollutants. So these are these PBDEs, PCBs, and organochlorine pesticides. And then we're also looking at the home environment, some information about the dogs and demographics and their environment from their annual surveys. And then we're taking these blood samples and we're going to analyze them for these chemicals. The other thing we're doing is we're working with the Dog Aging Project, which is a long-term study that's planning to follow dogs for 10 or more years, and it's based on citizen science and open science. So the data gets published and is available. The goal is to understand how the influences of genes, lifestyle, and environment impact aging. And ultimately they want to understand how to increase human and dogs' healthy lifespan. So increasing the time that we have with healthy life as opposed to having more diseases later in life. This cohort has more than 50,000 dogs of all sizes, breeds, ages across the United States. And the study's ongoing. So if you Google the Dog Aging Project, if you haven't already, you can enroll your dog, and you have annual surveys that you'll complete. And then additionally, there are other various nested studies, and they are also developing a biobank. Our current study is, again, a nationwide study, and we're working with Dr. Audrey Ruppel at Virginia Tech. And that we have 76 dogs, and of those dogs, 38 of them live in rural environments, and 38 of them live in urban environments. And it's kind of split evenly between the size of the dogs. And all of these dogs wore those silicone samplers for 5 days during the month of August in 2023. And then we also have access to the home environment and dog demographic data, and we're doing the same type of screening for chemicals in the silicone dog tags. Overall, what we hope to achieve with all of our research is to identify modifiable factors for cancer and other health conditions, because we'd really like to aid in prevention of cancer. we like to provide scientific evidence so that we have safer products and better policy decisions about chemicals. And we want people and dogs to live longer and healthier lives. So one of the big questions is, how can you minimize your dog's exposure? Well, your everyday actions and choices can have an impact, even if it's something small. Just making any small change can start to make a difference in your overall exposures. So one thing is minimize the use of household pesticides and herbicides. If you can avoid it and you need to use lawn treatments, then keep your pets off the grass for an extended period, at least 24, if not 48 hours after it's been applied to the lawn. Also, if you see these caution pesticide application signs in public places or on other people's lawns, try to avoid your dog walking on those. Choose your pet products carefully. So looking for labels on things like beds, toys, and other items. So you can look for things that say phthalate free or PFAS free. But the other things you can look for are these Green Guard Gold certifications or these Oikotex certifications. So these are these little green tabs on these photos that just have a certain level of certification of being free of certain chemicals. The other thing you can do is vacuum and dust regularly. Many harmful chemicals can accumulate in the dust, which, especially with the grooming behaviors that our pets have, they can also ingest these chemicals. And dusting can just be simply wiping with a damp cloth. It doesn't have to involve more chemicals. But speaking of more chemicals, choosing safer cleaning products or personal care products. So avoiding air fresheners and heavily scented products, because these can release volatile chemicals into your home. And one label you can look for on products is this EPA Safer Choice label. So pets truly are a sentinel species for health. Our health is their health. Our exposures are their exposures. They really are our man's best friend. So I'd like to thank all of the people that are involved in this. As you can imagine, there is a whole team that goes into this, including Dr. Heather Stapleton and all of the people from her lab, Dr. Matthew Breen from NC State University, the people that fund this type of research, such as the Canine Cancer Alliance, the Morris Animal Foundation, the V Foundation, the Consortium for Canine Comparative Oncology, all of the people and their pets that participate in these types of studies, all of our collaborators from the Dog Aging Project, especially Dr. Audrey Ruppel, and then other people that we've worked with from NIH and the CDC. So thank you for your attention, and I guess we may or may not address questions, but I know some of you submitted some questions, so I did preemptively prepare a few things just that hopefully address some of the questions that were not already addressed. So just a disclaimer, I am not a veterinarian. I cannot provide any specific medical advice, and the following are just general suggestions based on the scientific evidence that's available today. So one of the big questions that a lot of people's questions alluded to was, can we prevent hemangiosarcoma? How can we reduce risk? Well, there's really no way you can guarantee prevention of hemangiosarcoma right now, but there are some things you can do to support your dog's overall health and reduce their risks in general. Aim to minimize exposures to environmental chemicals, such as the slide that I had previously presented. Keep your dog active and at a healthy weight, and keep up with routine veterinary wellness and testing. This helps track baseline and trends of your dog's health over time. How much of this disease is genetic versus environmental? It's both. George Bray, who was a researcher focused predominantly on obesity, he said, genetics loads the gun, but the environment can pull the trigger. So while you do have some of these breeds that carry genetic risk factors for hemangiosarcoma, not every dog with these genes develops cancer. Environmental pollutants and even hormones can influence the risk of exposure. So an example is, think about smoking. We have an abundance of evidence that smoking is associated with lung cancer. But, I'm sure many people know somebody who was a lifelong smoker who never developed cancer. And it's this complex interplay between genetics and environmental factors that ultimately impact risk. And that's why studies such as the Golden Retriever Lifetime Study and the Dog Aging Project are so important, because they can untangle how these different factors work together. What tools for early detection and treatment are there? There's a lot of ongoing research using cutting edge technologies today. A lot of it's focused on detecting hemangiosarcoma earlier and more accurately, and some of these methods are using ultrasounds and CT imaging, but we're also seeing people that are trying to do AI and machine learning tools to help with these diagnostics. There's also new treatments that are being developed to improve survival in some dogs, such as targeted therapies, immunotherapies, and vaccines. So how can you help? Well, if you participate in studies like these or registries with your dog, it helps researchers like us try to find answers. You can also support organizations that fund cancer research. And then also just staying informed. So seeking out science-backed information and sharing that information with others. Again, thank you for your attention. And if we have any time for questions, happy to try and answer some.
Yeah, thank you so much. So obviously I have a concern because last year we lost our English Bulldog to lymphoma. She was a grand age for an English Bulldog, but still it was lymphoma. Now this year my German Shepherd just had his spleen removed 2 weeks ago. We got the lab results last week, or it's HSA. Is there any place we can reach out? I mean, we do all natural cleaners and all of that stuff, but is there any place we can reach out and find out if there is an environmental impact? 2 dogs in 2 years. This is concerning for me.
Yeah, I mean it's a challenge, and unfortunately it's a story that that we hear a lot. And I mean, there's no clear-cut answer right now. I personally am not doing anything specific with lymphoma research. I know there are groups that are doing that. I know there's a group that is looking specifically with this Golden Retriever lifetime study, looking at environmental factors associated with lymphoma. I know that some of the data is again related to some of these pesticides specifically. 2,4-D is one of the chemicals that has been published to be associated with lymphoma, and just reducing things like paints and solvents. Those were some of the things that have been found to be associated with lymphoma. Can't point you towards any specific studies for lymphoma that anybody's recruiting for. But like I mentioned, that just those, trying to reduce overall exposures right now is kind of the best place to start.
So one thing that
Thank
you
you.
might look into, is improving your dog's gut microbiome, because sometimes these exposures can end up dysregulating the immune system. And one of the ways that happens is through the gut. And there isn't a study with canines, canine cancer yet, but based on human studies and mouse studies, just having really good, diverse, healthy gut microbiome does help. So before your dog gets cancer, or even after your dog gets cancer, giving them fecal transplant. There are these fecal transplant pills that you can buy from some companies. And again, there's no definitive data yet, but based on what's known in humans, it may not hurt, and it may even help to just start improving your dog's gut microbiome. So that's actually another study that we're going to be starting very soon, looking at the gut microbiome and how dogs with cancer does have dysbiosis, dysregulated gut microbiome, and how improving that might help them fight cancer. For example, improve their response rate to immunotherapy. So that's something that we'll be talking about in future webinars and resources. Thank you.
Thank you very much.
Hi, my name's Mo. I actually have a question in relation to hemangiosarcoma. My question is, as we are actually taking these proactive measures to actually do, have a trend of the vitals, do perhaps like an annual blood test, when it specifically comes to hemangiosarcoma, what are these trends that you're kind of seeing that there's a variation in? Because I was wondering how often would you want to get imaging such as like an ultrasound done on a canine, or when that triggers that, right? I imagine that the vitals will actually show, hey, here's where we should actually probably visit maybe an ultrasound at this point. Um, so yeah, my question kind of goes back to what are those tiny preemptive moves to actually say, hey, the doctor was talking about the blood panel showing these in the results. I think it's a good idea to actually start looking at some additional imaging. Is there perhaps maybe a recommended screening cycle that's like recommended for dogs or canines, when it comes to things such as more expensive imaging, such as, x-rays, perhaps more importantly, the ultrasound?
So I think this is a really good question for veterinarians and veterinary oncologists. And that I do know that there is a study that has just been completed, which is a preventive vaccine using something called frameshift peptide vaccine that was invented at Arizona State University. And they had a very large study giving this vaccines that could potentially prevent or reduce the risk of getting cancer to dogs. And they've been monitoring these old dogs for several years. And one of the things that they did share was that they realized that because of because these dogs were part of the clinical trial, they were going to veterinarians much more frequently, like 2 or 3 times a year, and that helped them catch cancer much earlier, which put them in a better position of getting treatments that actually worked. So there isn't a really like definitive study, but in future, we hope to invite a veterinarian or a veterinary oncologist to talk about this. Another point that I think we should mention, a lot of people asked about, is are there tests to do early detection for hemangiosarcoma? And hemangiosarcoma, unfortunately, well, there are many tests now: urine test, saliva test, blood tests.
I was behind the trash truck.
But most of these tests, some of them are even home tests that you can just send in urine samples and urine samples and saliva samples from home, and they're quite affordable. But the problem is there are very high false negative rates and also false positive rates. So you could get a result saying, no, yes, your dog has cancer, but after you do a whole bunch of other tests, you realize, no, he doesn't have cancer, and reverse. And I personally experienced this many times. So you have to be very careful when you choose these tests that are now being advertised online, because there isn't really great data behind them.
Thank you, Dr. Wise. Thank you, Mary.
I have an interesting scenario. I have 2 golden litter mates. One was diagnosed with sub-Q hemangiosarcoma, one was not. That was 3 years ago. So same environment, same genetic makeup, and one was diagnosed and one was not. And both have been scanned pretty regularly. We're fortunate enough to have his oncologist through the Purdue Vet School.
Yeah, so that comment about how genetics loads the gun, but the environment pulls the trigger. While they do have shared genetics, they're not necessarily identical, and it's just this complex interplay between how the environment impacts each specific gene from each individual, that we just don't fully understand yet. Some of one thing I can point to about how different and different environments and different genes. You know, we look at twins who have the same exact genetic material, and you know, there's the example of the twins where one went to space and one stayed on Earth, and you see this massive difference in just their appearance based on that. It's something that we just don't understand enough to be able to explain why you know, one develops cancer and one doesn't, even though they're related, they have similar genetics, and it's because there are small things in the environment that you know, while they live together, may be slightly different in terms of how much one dog, you know, grooms versus the other, or whatnot. It's really complicated, and you know, we're hoping that, you know, as these data progress from these really large scale studies, that we'll really start to have a better understanding as to, you know, the how and why.
That's right. Littermates don't have identical genetics. That's an important point. So I should remind you that this study, as well as the EGFR HER2 vaccine and other immunotherapy clinical trials that we're supporting, they're all made possible by generous donations of supporters like you. So please consider donating to Canine Cancer Alliance so we can continue these studies. And we actually are now accepting picture submissions for our calendar for 2026. It's called Love Calendar. It is probably the most beautiful calendar you could ever get. So, and we'd love to have your dog or cat's photos in the calendar. So check out our newsletters, our email blasts, so to find out more. I do have a question, Dr. Wise, about dusting. You mentioned the importance of dusting now. Can you explain, can you help us understand why that's important?
Yeah, so a lot of these chemicals that we're able to detect, such as, you know, the flame retardants and the pesticides, they can actually kind of off gas from the materials that they're in, and then they can, you know, the ones that are, you know, particles, they can accumulate in dust. And Heather can step in if she wants to give, if she thinks I need to explain further, but they can accumulate in dust. So you both inhale dust, but also when you breathe in and you get dust in your mouth, you can ultimately end up ingesting some of it. And when we think about our, you know, our dogs and how much time our dogs spend, you know, on these, you know, surfaces like our, you know, on the floors where they're, you know, can be in contact with this dust, and then their grooming behaviors, that they're probably ingesting a lot more of this dust. So just being, you know, wiping these surfaces and removing the dust is can remove some of those chemicals from the environment.
Thank you.
I have a question on another environmental factor, which is kibble. There's been some rumors and anecdotal things about the way kibble is made in the high heat, that maybe the kibble is not the best thing to be giving dogs, and we should switch to a different diet. Is there any, you know, any evidence on that from an environmental perspective?
I haven't seen anything that's specifically identified anything in kibble as a risk factor for cancer in dogs. I will say that you know, I mean, not all kibble is created equal. Some companies, at least, they, you know, they do more rigorous nutritional testing compared to other. So you know, there are some companies that make sure that, you know, there is adequate nutrition and vitamins and the essential nutrients in kibble. And so it's a complicated balance because you need to make sure that your dog is getting proper nutrition. I would still say the best thing you can do is to speak with your personal veterinarian and speak up to what the best option is for your specific dog based on their, you know, both their breed but also their life stage, because you know, we want to make sure that we're still getting, you know, all those other important nutrition factors in their diet.
There's a question, is there any studies to understand how fast this disease progresses and if there is anything that can be done to help slow it down? So there is a lot of data published about how quickly hemangiosarcoma progresses. Unfortunately, it metastasizes very quickly, and it's a very abrupt disease. The good news is that we now have strong evidence that immunotherapy treatments do help. They do work. And so instead of just trying to kill tumors, cancer cells with chemotherapy, if you could activate the immune system in the right way, for example, using therapeutic vaccines like EGFR HER2 vaccine or tumor lysate vaccine is another vaccine that you can make if your dog has splenectomy and has a tumor removed, they could actually process that tumor and turn it into vaccine, inject it into the dog, educate the immune system as to what cancer cancer proteins look like, and actually stimulate the immune system in the right way. And there was actually a really beautiful randomized trial that took place about 30 years ago with a drug that consisted of miramil tripeptide. It's another form of immunotherapy that showed that it could really dramatically extend the survival time for stage 1 and 2 dogs with hemangiosarcoma. But unfortunately, it's not available for dogs today. We are looking into it and trying to see if it could be made available, either manufacture it or to get some research grade of this drug and make sure that it's pure, et cetera, and make it available. But there is very, very strong evidence that immunotherapy can help. So stay tuned because science is moving very, very rapidly. Okay. There's a comment about University of Helsinki's research showing that inflammation markers increasing on kibble. Yeah, that might be true, and it depends on kibble. There's so many different types of kibble. I know many dog parents switch to homemade food, etc., when their dogs are diagnosed with cancer. But I think there is now more research looking at gut microbiome. So stay tuned on that. We did have a lot of questions about early cancer detection, early hemangiosarcoma detection. So we will probably have another webinar on this topic. Is there anything else? Well, I had one more question, Dr. Wise.
Mm-hmm.
In your earlier study with bladder cancer, what was surprising to you? Was there any chemical that really stood out as being extremely harmful that's linked to cancer? Or was it like broad range of chemicals that dogs were exposed to? What was surprising?
I mean, I would say it's definitely, you know, that one graph that it showed where you have all the different different dots was each chemical, and just the amount of chemicals and how much more chemical exposure that dogs with with bladder cancer had compared to the healthy controls. Because in reality, we are exposed to so many chemicals, and it's this cocktail of chemicals in our environment that we're exposed to that are likely contributing to the development of disease. It's not necessarily one bad actor, it's several bad or moderately bad actors that when you put them all together, act in different ways. So one could be acting on the immune system, and one could be acting on the microbiome, one could be impacting, DNA damage. And when you put all of those together, that's when you get that that result in cancer. So I don't think it's necessarily one bad actor. It's this chemical cocktail that's ultimately contributing to disease.
Got it. Thank you. And I'm going to make a comment about the beginning of your talk. You began by talking why it was so important to study dogs. And I know that for this audience, it's obvious because we want to cure cancer in dogs. We want to prevent cancer in dogs. We love people too. But
yeah.
I think listening to you, it made me now start to worry that I should be worrying more about people too. Yeah, thank you so much. We will follow up with the transcript and recording of this video, and we'll probably create notes version of it and share it with you. If you have any comments or if you have any ideas for other future webinars that you'd like to listen to or for us to dig into, let us know. We do tend to focus more on treatments because treatments are easier. Actually, it is developing better immunotherapy treatments that works, I think, is within reach. I really feel that from talking to different researchers. But risk reduction, prevention is extremely important, and it is harder. It's actually a harder problem to solve. And sometimes I feel like, oh my gosh, it's overwhelming. It's too much. It's just everything. But we shouldn't feel like that. I think we should feel empowered and learn as much as possible how to help protect ourselves, protect our dogs from harm. So it is just as important. So thank you so much, Dr. Wise, and thank you, Dr. Stapleton. And we're really looking forward to learning more from your study. It's going to be a really exciting 12 months as you uncover what's going on. And if you would really like to support this study or want to see this study expanded or support us in any other way, please reach out to us or just send pictures of your dogs and become a monthly donor. We have special monthly donor webinars and behind the scenes information that we share. So thank you so much. Thank you, Dr. Wise. Thank you, Dr. Stapleton. Thank you, everybody, for joining us.
Thank you.